Current strategies for urine drug screening are expensive to perform for routine analysis of increasingly large panels of drugs of abuse, as required for workplace testing, as well as therapeutic drug monitoring applications. Additionally, methods based on immunoassays are prone to bias and are ultimately limited to target analysis of known drug panels.
The main advantage of our method, which is referred to as MSI-CEMS, is that it allows for the rapid screening of an unlimited drug panel and their metabolites with high accuracy and minimal sample workup.
There is an urgent need for a high-throughput and affordable screening method to improve patient outcomes by confirming adherence and optimizing drug dosage regimes while revealing illicit drug usage. This multiplex separation platform can be applied to a variety of other research areas related to targeted or non-targeted metabolite profiling in complex biological samples.
Begin by thawing de-identified morning urine samples collected from patients with a known prescription drug history from -80 degrees Celsius storage on ice and vortexing the thawed samples for 30 seconds. Next, sediment the samples by centrifugation and combine 3 10 microliter aliquots of each sample with 10 microliters of deuterated internal standards, 5 microliters of a 4-fluoro-L-phenylalanine and 3-chloro-L-tyrosine solution, and 25 microliters of deionized water.